In the vast land of Bharat DNA Pradesh, there lived a farmer named Genome Baba, who was responsible for growing the crops of life. These were the genes that decided everything about living beings. He had a huge field called the human genome field, where each crop represented a specific gene. One day, Genome Baba noticed that some crops in his field were diseased. These faulty crops caused genetic disorders, low immunity, or even undesirable traits in people. Baba wanted to fix these problems, but didn't know how to remove the diseased crops without harming the healthy ones. Then came Dr. CRISPR, a scientist from Bharat Vigyan Kendra, who brought an amazing genetic tool with him. He had also hired a sharp helper, cas9, who could do precise work. Together, they promised to fix the genetic field. Genome Baba pointed out the diseased crops in the human genome field. These diseases included genetic disorders like sickle cell anemia or inherited diseases like thalassemia. Dr. CRISPR used a guide RNA, a kind of map, to identify the exact location of the faulty crop in the vast field. Dr. CRISPR and his assistant Cas9 walked into the field. The gRNA acted like a tracker, leading them straight to the faulty crop. They didn't disturb any of the healthy crops while finding their target. Once they reached the diseased crop, Cas9, the expert molecular scissors, carefully cut it out. This step removed the faulty gene that caused the problem. After removing the faulty crop, Dr. CRISPR planted a new, healthy gene in its place. This new gene ensured that the problem would not occur again, and the field would thrive. After the repair, Dr. CRISPR monitored the field to ensure that the new crop grew well and integrated seamlessly with the rest of the field. Genome Baba was thrilled with the results. This technique, CRISPR-Cas9, could now be used across Bharat DNA Pradesh for various purposes: curing genetic disorders, improving agriculture, and fighting diseases.